Okay, seniors, this is your requested review screencast for topic 6 and 11. This is, as you remember, a huge unit, so I'm not going to go through everything, of course. What I'm going to do is, by request from Luca and Amr, I'm going to go over neurons and synapses, respiration, and movement for HLs. So, let's go to respiration first. Blood system, heart dissection, infection against diseases. That's the HL1, antibodies, vaccinations, very relevant right now. Gas exchange. Okay, so this is by Luca's request, gas exchange. So if you are interested in this one, then watch. If not, just kind of skim through the video to the next title slides. You can see what you're interested in reviewing. So here's our long list of things for this subtopic, talking about ventilation, different types of pneumocytes, how air is moved through the respiration system, which muscles contract to do this. We'll talk about lung cancer and emphysema, different muscles working as antagonistic muscle pairs, and ventilation, which is practical 6, which you guys have done. So first thing, we need to have oxygen for respiration at our cells. Every cell has mitochondria, which is every cell, needs oxygen to do that, and produces carbon dioxide as a waste product. We have to get rid of that. So to do that, we're constantly filling up our lungs and expelling our lungs, filling them and emptying them back and forth all the time. And that air follows a very specific path, right? It's going through the nasal cavity usually, down through the pharynx, past the larynx, into the trachea, and then branching into one lung or the other through the bronchi. It gets the little tiny bridge called bronchioles, and those end in alveoli, these little sac-like structures that are very well designed for gas exchange. So the function is to get oxygen in and CO2 out. It's going to happen through the alveoli and the capillary beds on top of them. This is a really important connection between the circulatory system and the respiratory system. Is we have these capillaries, because they're single cell thick blood vessels where we absorb nutrients in the digestive system, where we do gas exchange in the respiratory system, where things can move in and out really easily. Okay, so here we have the alveoli, very sac-like structures covered in capillaries. There's two types of alveoli cells. There's type 1 pneumocytes, which are these very thin, kind of bubble-like cells we traditionally think of as the alveoli, and they're the ones that where gas exchange actually happens. And a really important factor here is they are not capable of mitosis. So if you lose alveoli from something like emphysema or lung cancer, you can't get them back in the same kind of amount because they can't make copies themselves. The type 2 pneumocytes are these kind of ones down here. They're very small, and their function is to secrete mucus. It's really important that the alveoli don't stick to each other. They're lubricated when they collapse and they kind of get smaller and bigger, expand, that they're able to stay separate and have that high surface area for gas exchange and don't get damaged. So the mucus produced by these type 2 pneumocytes do that. These ones also can do mitosis to make type 1 and type 2 pneumocytes. The reason why we don't have as many of them, as many alveoli, if we have this issue, if we have like emphysema or lung cancer, is it can't replace all of them. So instead of having like 15 or so little alveoli here, you get one big one. It's much less surface area for gas exchange. Okay, so what muscles are happening for inspiration and expiration? Well, inspiration, we are making the thoracic cavity as big as we possibly can to decrease the pressure. So to do that, we're going to lower the diaphragm, and the diaphragm lowers when it contracts, and you can feel that by actually trying to change your diaphragm. How does it go down and up? And you can feel when you relax, it goes up. It takes a little practice to feel it, but if you're a swimmer, most kind of athletes can do it. If you're a singer, any kind of musician, you can probably do it as well. The intercostal muscles, the external ones, are going to contract, and the diaphragm contracts to create a big space. The intercostal muscles are going to relax, the externals are going to relax, but the internals are going to contract, and the diaphragm will relax, and abdominal muscles will also contract. The abdominal muscles don't often contract because they're not needed for this, but if you're doing really deep breaths, it will happen as well. And notice how the size of the thoracic cavity here is really big, and here it is very small. Okay, so take some deep breaths and feel what's happening. Remember always, your lungs don't move. We're not sucking in air and pushing in air through our lungs. We're creating different space for the lungs through the muscles surrounding them in the thoracic cavity, and that's causing the air to come in and out. Because a change in the size of the thoracic cavity changes the pressure, and that creates a pressure gradient. So when we have these muscle pairs working together to create this different size thoracic cavity, we're creating different pressure gradient. So here are the two pairs, as we have the diaphragm and the external intercostals. Those are always working for inhalation. So external intercostals and internal intercostals, those are antagonistic pairs. And then the diaphragm is paired up with the abdominal muscles. They do opposite things. Remember, muscles can only pull, they can't push. They have to have a pair to the opposite motion. Okay, so those are muscle pairs in ventilation is the diaphragm with the abdominal muscles, the external intercostals with the internal intercostals. Those are our sets of pairs. Okay, so remember, review this. This is a really helpful diagram. It shows that if we increase the thoracic cavity size by lowering the diaphragm and expanding your ribs, you're going to increase volume, which means a lower pressure, which means air will naturally move in from the higher pressure in the air around you into your lungs. If you decrease the volume, but your ribs move in, your diaphragm moves up, that's going to increase the pressure by making it less volume, and that will mean the air will actually move out towards a lower pressure in the air around you. Okay, so here's our two key ideas. The structure of lung tissue is ideal for its function of gas exchange and with thin-walled alveoli lubricated by type 2 pneumocytes, and cartilage to keep the trachea open but flexible. So the trachea, you can feel the cartilage rings on it. So another great example of structure and function in the respiratory system. Inspiration and expiration are the result of pressure changes created by lung volume changes. The lungs are given more or less space by the diaphragm and intercostal muscles. So we measured ventilation with different spirometers to do this. Different types of spirometers we used to check on the peak flow, kind of how much gas is coming in and out of your natural breathing rate. Emphysema. So emphysema tends to happen from tobacco smoke. Long-term marijuana smoke can also do this. Manufacturing fumes, coal dust, air pollution. And what happens is your alveoli that are normally healthy like this become these big hole-like structures. Here's a lung tissue that has emphysema. And those large holes, means you have less alveoli, which means you have less surface area for gas exchange. Now, there's no cure for emphysema because you can't undo it. You can't, those cells cannot go through mitosis. So the only way to treat emphysema is with a lung transplant, which is not a transplant, it's not a cure. You're just replacing the organ with a better functioning one. Okay, lung cancer. Cancer is always just a growth of cells, an uncontrolled growth of cells. So in lung cancer, it's happening in the lung cells. This is a very prone cancer to metastasizing. It moves from one location to another in the body. Okay, that tissue, so this part of the tissue here has a tumor, that's no longer functioning. Those alveoli can no longer do gas exchange. And if you remember, we need to have our alveoli and for gas exchange, and if we get rid of too many of them through emphysema or lung cancer, we're going to have shortness of breath and perhaps trouble breathing. This is caused by carcinogens, which are cancer-causing agents, which can enter the bloodstream and into the lungs, and tobacco smoke is one of the number one cause of this. Look at some data here for how the smoking status affects the age at which cancer happens or doesn't happen in individuals. So the full solid lines are never smoked, male, female. These are the dotted lines are current smokers, and these ones are former smokers. Okay, key ideas. Since type 1 pneumocytes cannot undergo mitosis in adults, any alveoli that burst due to emphysema or are lost due to cancer cannot be replaced. Lung disease results in a significant reduction in the surface area for gas exchange. The number one cause with lung cancer and emphysema is tobacco smoke, although other factors, both genetic and environmental, can also lead to these types of lung disease. You do practical 6, a good practical to think about, especially for consent and how you have to seek that, and the controls and constants you have to try and be aware of when you're working with human subjects, and confidentiality as well. Okay, and then the next request was to do neurons and synapses. So let's talk about neurons. Let's talk about electrical impulses moving through as we signal things along the body. So we have the nervous system. It's composed of two main parts. We have the central nervous system here, the brain and spinal cord. Oops, come back. Okay, the brain and spinal cord. Peripheral nervous system is all the nerves, all the body. They have to work together to make this whole thing work. So we have to have the brain to collect the data and send signals out, kind of compare the data to set points and see if I need to react in any way. The nerves are going to collect a lot of information about what you're feeling and also send signals to react, different muscles, different cells, different organs all over the body. Okay, so we have the autonomic, the self-controlled things you're not thinking about, and the somatic skeletal muscle, we're actually constantly thinking to move those things in the nervous system. Okay, so we talk about neurons having different parts. The main three parts of neurons is they all have dendrites, where they're going to collect signals from other neurons. You have the cell body, which has the nucleus and has the mitochondria, helps keep the cell alive, and you have the axon, which is this part here, where it's going to be sending the signal down to the next neuron, is the action potential. Okay, so key ideas: the structure of the nervous system allows information from various sense organs to be processed and responded to in an efficient manner. While there is some variation in the structure of different types of neurons, they all have dendrites connected to a cell body leading to an axon. So I had you guys color in your large foldable, so I'm going to just summarize the key ideas here. You do have notes on this. If you have any questions, please email me, ask me in Zoom chat, stuff like that. So we have the dendrites, which is the end part here, branching off to collect signals from other neurons. We have the axon hillock right here, where you have to reach that minimum threshold action potential of negative 55 millivolts. And you have the Schwann cells, which are here collecting and wrapping around the axon in a myelinated neuron, helping go faster. The axon terminals, okay, so the axon terminals down here, that's the end of the axon, the kind of buttons where it will send a signal across the synapse to another neuron. You have the nodes of Ranke, which are little gaps here between the myelinated part. That's going to be where the signal will jump to if you have a myelinated neuron. And the other one was the cell body. So here, where you have the nucleus and the other organelles you need to have to keep that cell functioning. Okay, the myelin sheath, okay, will be the entirety of all these little Schwann cells together along the axon, and that allows, if you have a myelin sheath, then you can do much quicker signals through saltatory conduction, and that's something that happens usually in really long or really important neurons, like the neurons that go from the base of your spine to your toes. Those are going to be myelins, so very long distance they have to go. Okay, and the other one here is the axon, which is connecting the cell body to the terminal buttons, and that's where the impulse travels down. Okay, we want to make sure you guys didn't mix up the difference between a neuron and a nerve. So a neuron is a single cell, and nerves are made of many, many of them. Here's all the axons of different neurons in a bundle, and the several bundles will make up one nerve. Okay, there's about 214 really important key medical or like largely well-known nerves in the human body, but there are way more that are different to people or not quite as important for key structures. We don't have a solid number of nerves in a human, but there are estimates go anywhere from 1,000 nerves to 1,000,000 nerves, depending on who you ask. Okay, key idea: the neuron structure allows it to perform its function of collecting signals from other neurons and efficiently sending an electrical signal down the axons to terminal buttons. Nerves are made of many neurons. While there are 214 medically important nerves, individuals contain many more nerves. Okay, let's talk about how the signal actually moves. So, when a nerve is at rest, we say it is polarized, and it's at resting potential, which is negative 70 millivolts. Okay? So the net charge, these charges we're talking about, is because we have the sodium and potassium ions, as well as these big proteins that are inside. So we have sodium on the outside and we have potassium on the inside. I always think of it as a salty banana, salt on the outside, potassium on the inside. Sodium and potassium from salty banana always helps me remember. And the difference here of being negative 70 inside is because even though I do have a potassium inside, there's these big negatively charged proteins that it doesn't have enough potassium to surpass and move the overall charge, the net charge, towards being negative, to being positive. Okay, so the normal kind of charge is this negative 70 millivolts. Okay, so for it to become polarized and maintain this resting potential, it's going to have to use ATP and active transport to use what's called the sodium-potassium pump. So they're going to pump 3 sodiums out and 2 potassiums in for 1 ATP, which is pretty efficient use of energy. So that will cause you to have the salty outside, the sodium outside, and the potassium inside. It's a way of resetting after the active potential moves down the neuron. Okay, so how is the new impulse going to move? Well, when it becomes positively charged inside the axon, like here, that's called depolarized. That is the signal. So here in the neuron, in the axon, this is the depolarized region, and most of it is still at rest. But that depolarization will still be moved down and down along the axon towards the synapse over here. After a part is depolarized, it's going to go back to being repolarized after, going back to negative. That takes a bit of time. And then back to rest. Okay, so how does this work? Well, the neuron's going to receive signals from other neurons at the dendrites, and that impulse is going to move through the axon. And that impulse, we just said, is a positive charge in the axon. That's what makes the signal. To make it reach at least plus 40 millivolts, that's our signal here is plus 40 millivolts, we have to have sodium ions moving inside. Remember, sodium is outside, salty banana, so they tend to move in through the sodium channels. And to do that, it has to open these certain channels. Now remember, to maintain this normal balance of mostly sodiums outside, potassiums inside, we're using this sodium-potassium pump to kind of maintain that. And that's our normal. So we are always doing that. And even if we get a pseudo, we're still doing it. But then we're going to be focusing on changing the charge very quickly to send that information along the axon. Okay, so once part of it is depolarized, it's going to become positively charged inside, and that will continue to move further down the axon. It's a chain reaction. So how this works, we have to have our negative 70 here is our resting potential. We have to have enough of a signal happening at the axon hillock to cause it to go to negative 55. So it's a plus 50 millivolt difference here. If you get a signal, but it's not strong enough to actually send it up to negative 55, then it will not happen. So here's a couple of failed initiations. It never really happened. Didn't get strong enough signal, which is good. You don't want your body reacting to every single signal it receives. That'd be crazy. So when you reach negative 55, the sodium gates are going to open, and then it's going to cause a huge influx of sodium into the neuron, and we're going to jump really quickly up to plus 40. Okay. So we have sodium rushing in, go from negative 55, so for sodium channels to open, they rush in, goes all the way up to plus 40. And that's the signal. Now, once we reach plus 40, that's a signal for the voltage-gated channel for the potassium to open, and the potassium moves from the inside out. So remember, here we have sodium moving in at negative 55, potassium moving out at plus 40. And that's going to cause it to move back to normal by moving all these sodiums are out and potassiums are in. That's the opposite. Sorry, potassiums are out, sodiums are in. That's the opposite normal. So then to get it back to normal after that signal has happened, then we're just jumping way down here to get back up to normal like this part of the curve. We're going to be using the sodium-potassium pump again to bring everything back to its happy normal place. Once it's been repolarized, everything's back to negative 55, negative 70 resting. Then it's ready to receive another signal. This happens absurdly quickly in cells. Okay, so key idea is the resting potential of an axon is negative 70 millivolts, and its sufficient signals builds in the axon hillock to pass negative 55 millivolts, then an action potential is initiated. During an action potential, the axon becomes positively charged 1 bit at a time through the flooding of sodium ions. To move back to rest at negative 70 millivolts, the potassium ions flood out of the axon, and the sodium-potassium pump restores the balance. So it's important you have the numbers, the different charges, and exactly what is moving when. Okay, so here's a nice summary diagram. So we have our normal resting potential, salty banana, sodium outside, potassium inside, negatively charged inside, potassium outside. The action potential happens, I reach negative 55, sodium channels open, sodium rushes in. That makes it positive inside and negative outside. Then I reach my plus 40, and my potassium is going to open, and potassium goes out, making this going back towards negative, and this is positive. And then to maintain my resting potential and kind of get back to exactly my negative 70 resting potential, I'm going to have 3 sodiums going out for every 2 potassiums coming in, restoring my kind of salty banana equilibrium. Another example here is a good one to look through this image. It shows the graph, the oscilloscope tracing, as well as the images. Okay, saltatory conduction occurs when you're going to be moving the cells, moving the signal down the axon very quickly. Instead of changing the polarity of the entire axon and sort of depolarizing the entire thing, you're going to jump the charge from one gap to another gap to another between the different Schwann cells, at the nodes Depolarization. You're going to move it bit by bit by bit. And that makes it a lot more efficient and quicker to send a signal. Okay. So myelin acts as an insulator, holding onto the charge at one end, so it can be passed on to the next end over here. Okay, and key idea, saltatory conduction allows for more efficient conduction of electrical signals down the axon by allowing the action potential to jump from one node Depolarization to another. Okay, that was the axon part. We'll do synapses next. So 6.5 synapses. So this is where we're taking our electrical signal we just talked about in the axon and turning it into a chemical signal here across the synapse. So here is the synaptic button or terminal button of one neuron right here, and here is the dendrite of another neuron. And this tiny gap between them is the synapse. Okay, so that signal is being sent by different neurotransmitters. So we have some examples here: adrenaline, dopamine, endorphins, acetylcholine. This is the one we want you to focus on for the IB. Okay, so again, this is the second half of your big foldable, so make sure you use it to help you keep track of everything. We're going to walk through the steps. So I'm just going to do it on here. So the first thing happens is the action potential arrives at the terminal button, and that charge, that change, is going to cause the calcium voltage-gated channels, that plus 40, to cause calcium to enter the cell. Terminal buttons. When calcium enters, it's going to cause these vesicles here that are full of neurotransmitters to react and move towards the membrane here, the edge of the terminal button. They will then fuse with the membrane, like we have here, and release the neurotransmitter. It could be acetylcholine, it could be dopamine, it could be adrenaline, some sort of neurotransmitter. Again, I want you to focus on acetylcholine. It's going to release the neurotransmitter into the synaptic cleft, the space between the terminal button here and the dendrite here, and they're going to bind to a receptor, a certain receptor protein that only works for that particular neurotransmitter. So as acetylcholine, this is an acetylcholine receptor, and that will then cause a signal to move through the postsynaptic neuron here, through building up in the axon hillock, and maybe still moving down the whole thing if it's strong enough. Now, we don't want the signal to last forever, that'd be bad, so instead we have different ways of taking that up. We have the reuptake transporter that can take up different neurotransmitters and to absorb them back in. We also have an autoreceptor that keeps track of the concentration of neurotransmitters in the cell to make sure it's sufficient to actually cause the signal to happen. And then you also have enzymes. For acetylcholine, is called acetylcholinesterase, which then breaks down the neurotransmitter to a non-functional subunits, and then they can be taken back up and then reassembled here. It's a quicker way to make sure they just stop sending the signal. Okay, so all those steps are here in the notes. Okay, the application of this, let's just go over these. So the key ideas: when the action potential reaches the synapse, calcium channels open, triggering the release of a neurotransmitter into the synaptic cleft. This neurotransmitter binds to the receptor on the dendrite of the next neuron. And then autoreceptors keep track of the amount of neurotransmitter in the synapse. Enzymes may break down neurotransmitters, and/or neurotransmitters may be taken up by reuptake transporter proteins. That's how we're controlling how strong and how long the signal lasts. So a really cool / terrifying application of this is neonicotinoid insecticides. And what they do is they are going to bind to the acetylcholine receptor proteins. So on the receiving neuron here, we have little proteins that normally receive acetylcholine signals, and then it causes it to react. But if you have a pesticide in place, that is designed to bind with that signal. But because the enzyme acetylcholinesterase here cannot break it down because it's not acetylcholine, then these are permanently filled, permanently sending the signal to the next neuron to its emotion signal. So since we can't signal to move, it's kind of constantly stuck these frozen and full receptors, the insect can no longer send signals to move, and it will eventually die. It's paralyzed and then dies. Okay, key idea: neonicotinoid pesticides use our understanding of neurotransmitters to paralyze and kill insects. The pesticide mimics acetylcholine, the neurotransmitter responsible for muscle contraction, and binds the receptor proteins on the dendrites of the postsynaptic neuron, blocking any further acetylcholine binding. We look at some nervous tissue in the microscope, and that's it. I'm going to do a separate screencast for 11.2 because that's only for HLs. Okay? Hope this is helpful for you guys.